Hi, everybody. This is Group 5, Caroline Kennedy, Kevin Williams, and Callie Plumley. For our final project, we decided to cover Chapter 15, DNA, the indispensable forensic science tool. The learning objectives for this chapter are to be able to name the parts of a nucleotide and explain how they are linked together to form DNA, understand the concept of base pairing as it relates to the double helix structure of DNA, contrast DNA strands that code for the production of proteins with strands that contain repeating base sequences, explain the technology of polymerase chain reaction and how it applies to forensic DNA typing, understand the concept of electrophoresis, understand the structure of an STR, understand the use of DNA computerized databases in criminal investigation, and list the necessary procedures for the proper preservation of blood-stained evidence for laboratory DNA analysis. While our presentation is just a brief summary of the chapter and we won't have a chance to cover all these learning objectives, we have highlighted the most important pieces. To begin with, we'll briefly explain what DNA is. The human body is composed of some 60 trillion cells, and within those cells are chromosomes. Chromosomes are composed of nearly 25,000 genes, which are the fundamental unit of heredity. These genes are composed of deoxyribonucleic acid, with each strand of DNA designed to carry out a specific particular bodily function. DNA is a polymer, which essentially means it is a very large molecule that is made by linking together a series of repeating units. For DNA, these units are called nucleotides. Nucleotides are composed of a sugar molecule, a phosphorus-containing group, and a nitrogen-containing molecule called a base. For DNA, there are only 4 bases: adenine, cytosine, guanine, and thymine. It is important to remember that the only arrangement possible in the double helix configuration of DNA strands is the pairing of bases A to T, adenine and thymine, and G to C, guanine to cytosine. These are known as complementary base pairings. In 1985, forensic scientists began applying DNA technology to human identity, initially focusing on tandem repeats of the genome. A tandem repeat is a region of a chromosome that contains multiple copies of a core DNA sequence that are arranged in a repeating fashion. The forensic community first focused its efforts on characterizing RFLPs, restriction fragment length polymorphism segments. RFLPs are different fragment lengths of base pairs that result from cutting a DNA molecule with restriction enzymes. While RFLP DNA typing has the distinction of being the first accepted protocol for forensic characterization of DNA, today short tandem repeat analysis is the most successful and widely used DNA profiling procedure in the forensic community. Short tandem repeats, or STRs, are a region of a DNA molecule that contains short segments consisting of 3 to 7 repeating base pairs. STR DNA typing is an essential investigative tool within the law enforcement and forensic community. The technology behind STR typing has progressed at a rapid rate in only a few years and gives vital evidence needed to resolve violent crimes and sex offenses. DNA evidence is crucial because it is impartial, implicating the guilty and exonerating the innocent. However, the importance of DNA analysis in criminal investigation has also placed great pressure on crime and forensics laboratories to improve quality assurance procedures and ensure the correctness of their results. The Combined DNA Index System is arguably the most significant tool to arise from a DNA typing program. This computer software was developed by the FBI to maintain local, state, and national databases of the DNA profiles of convicted offenders from unsolved crime scene evidence and the profiles of missing people. CODIS gives crime laboratories the ability to access these databases and match crime scene evidence to convicted offenders and criminals that are already in the system. In the last few decades, DNA profiling has catapulted biological crime scene evidence to a stature of nearly unparalleled importance. However, before DNA evidence can be used in the crime-solving process, it must first be properly collected and preserved. Before evidence collection begins, biological evidence such as bloodstains and other bodily fluids must first be photographed and thoroughly examined by an expert without disrupting the crime scene. Once this has been completed, the evidence collector should handle all biologically stained material with as little personal contact as possible. When packaging biological evidence, collectors must be certain not to package evidence in plastic or airtight containers, which can potentially accumulate moisture and result in the growth of DNA destroying bacteria and fungi. Each biologically stained article of evidence should therefore be packaged individually in either paper bags or well ventilated boxes with a red biohazard label attached to each container. For collecting DNA samples from victims and suspects, a buccal swab should be done, which entails vigorously swabbing the individual's cheek cells with a cotton swab, resulting in the transfer of buccal cells onto the swab. Occasionally, when an individual is unable to give a DNA reference sample, the needed DNA material can possibly be collected from items such as a comb, a toothbrush, or even earbuds. This chapter does a great job of illustrating the integral role that DNA evidence plays in the crime-solving process, as well as how much progress has been made in the technology behind DNA analysis in the last 30 or so years. Thanks to DNA analysis technology, not only are countless crimes solved quickly and efficiently today, but we are also able to solve older cases that previously went unsolved due to the lack of technological resources available when said crimes were committed. Although this has just been a brief summary of the chapter, I would highly encourage all of our classmates to go back and read this chapter, as it shows what a truly important role that forensics plays in the crime-solving process.